I took the charge of this as okay, what can I come up with that says what's cool about earthworms and why should you want to include them or turn students loose on them or get them started on something that you can do in your own backyard. So this is what I'll try to go over, a little bit of sort of, a little very, very brief introduction to the very diverse earthworms of Brazil. A case of Pontoscolex coryphurus, which is the most abundant, probably, earthworm in the world. And that has contributions from two colleagues, Luis Dupont in France and Luis Cunha, a Portuguese guy now working in Curitiba, Brazil. And then another one, an endemic or local regional species complex. And then a couple of other bits and pieces about diverse lineages that we've uncovered during a DNA barcoding project. So we know there's at least 320 species of earthworms in Brazil. This doesn't sound like very much, and we think this is a gross underestimate, and it'll become more obvious soon. I mean, I have a manuscript in my computer with another 20, and then there's the rest of the collection in Curitiba containing dozens more. And there's about 275 of those native, belonging to these four families, the Rhinodrilidae and the Glossoscolecidae, formerly known together as the Glossoscolecidae, but I've revised it. Octernodrilidae, an interesting and very, very poorly studied family, which turns out to be extremely diverse in Bahia. This we just discovered. Almadidae live in wetlands and are pan-southern tropical. They're on Africa and Asia as well. So that's kind of interesting. And then there's a whole lot of exotic species. Introductions, invasive species of various provenances. Some of them are Asiatic, and there's others that are African, and a few in southern Brazil in higher elevations there that are European. So like Colombo, Paraná, which was founded by Italians, we found some more or less Italian-looking earthworms living there behind a church at Embrapa Florestas. When we go around looking for worms, we find stuff that we didn't think was there anymore. Phimoscolex sporatocatus was described from the area adjacent to a motel south of Conselheiro de Lafayette. And if you're Brazilian, you know what a motel is, and you wonder why the earthworm guy was looking around the outside of a motel. Our Brazilians in the audience. Okay, good. So anyway, we found it, but not there. It was actually funny. We were wandering up and down the highway trying to find this motel, and we would ask guys, we better ask older guys. And they would say, oh, I remember when I needed a motel. Okay, onwards. Giant worms, Nyokusus. You will see signs along the road saying, Temos Nyokusu, because if you want to go fishing and you want big worms, you want giant worms. All right, so here's two Nyokusus from the Serra do Mar near Curitiba. And these were unknown until some students working in the lab in Curitiba went for a hike and found a couple of them mating on the path going up one of the mountains. This was taken by a British tourist on his way to Pico da Neblina on an eco trek. And Jose here is holding the bottom end of the worm is there. It goes up over this facão here, over a stick here, and back down to there. So even if this guy is really short, this worm is really long. And you will never dig one of these up. You just walk around in the forest on a rainy day, and then you will find it. This is what we politely call an earthworm cast. This is earthworm feces. This is in Amapá, on the way to São Lourenço in the interior, an old gold mining area, still actively gold mining. And the worm that did this is this thing. So there it is. It's 1.5 meters long. We got it out with a retro excavadora, a Fiat Alice with a cubic meter bucket, and in one scoop, it came up dangling a meter of worm from the teeth of the bucket. We pieced it back together. It got damaged. But this, look at this color, here and here. This is in a lowland tropical forest, sort of transitioning from cerrado on the coast into a more humid forest in the interior by somewhere. And so there shouldn't be dark soil like this, but I'll get to that later. Okay, floodplain earthworms. These are building biostructures. We call them, some people call them murundungs, we just call them condominios, because this is George Brown, my colleague and partner in Brazilian earthworm study. Every speck that you see in here that isn't a plant part or George is an earthworm fecal deposit. And this is in the seasonally flooded, but 6 months of the year, very dry coastal plain of Amapá, north of the Amazon River. This is in, I think, in Mato Grosso and the Pantanal. These are all piles of soil made by earthworms. And this way, they can have a relatively, they can be above the water line during the flooded period. So this is what they look like in the dry season. And I just have to point out that just not only in situations like this, but earthworms are the top ecosystem engineer in many biomes. If there's enough water to have green, leafy plants for several months of the year, there are probably earthworms in part of that region, maybe even in Caatinga. This is an totally understudied area as far as earthworms are concerned, because as the previous speaker said, hey, that's just a wasteland, you know, that's what everybody thinks. So why go there to look for worms? In addition to being major structurers of soil and nutrient cycling, earthworms are typically the dominant animal biomass, at least macroscopic animal biomass, of every system in which they occur. One French author said we know more about the kilogram or so of birds flying over and residing on any given hectare than we know about the 2,000 kilos of earthworms living in that hectare. So anyway, it's generally true that they are among the top animal biomass components, but they're out of sight and out of mind. Previously dry season, this is wet season. This is in this is actually in Colombia, but these are biostructures. These are built by, or at least maintained by, earthworms in a seasonally wet place that humans turned out to actually start exploiting these as places to have a garden. You can put crops on these, little patches here and there, and have them up above water level and able to survive and grow. Okay, so now I'm going to start talking about a specific example, which is Pontoscolex coryphurus. And these are slides provided by Lise Dupont in the University of Paris Est Créteil, which is one of the many Parisian universities. So here's a picture of a Pontoscolex, an unremarkable woodworm. It's known colloquially, or sort of by the population in rural areas of Brazil as minhoca mansa, the lazy worm, the slow worm. And this shows you where in the world it is known from. However, I think there's a few more records that the person compiling this did not have. But basically, you can just say that if there's a humid tropical soil, or even not so humid, it's there. So it's endogeeic, which means it lives in soil proper and consumes soil organic matter. We suspect, we think it's native from the Guianan Shield region, somewhere around in here, because all of the other members of the genus Pontoscolex are there, or somewhere nearby, maybe in the northern Amazon basin area. There's other Pontoscolexes, and it's hugely important for its effects, and it's highly invasive as well. And you can go around the gardens of this hotel, and I can show you where Pontoscolex are living, if you really want to. Our colleague Luis Cunha just published a study of the genetic diversity in the Azores Islands. So that's a long way from the Guianan Shield, but there's a pineapple growing business in the Azores, and so perhaps that's how they got there. But what he found is great genetic diversity within nominal Pontoscolex corythuras. This population is a lot of diversity, or well, little genetic differentiation within this haplotype cluster that lives in a volcanic field of sort of hot vents of steam and stuff like that. And these are tetraploids. And then there's a very great long separation. These are the 298 SNPs out of a 4,000 base section of mitochondrial genome. And then you have other sites in the Azores nearby where you have a lot more haplotypes and a diploid population of this worm. So, then we look down here. This is the tail end of Pontoscolex corythuras. And this will come back to you because this is one of the reasons why this worm is easy to work with, even if you don't know anything about earthworms, because there are very, very few earthworms that have a tail that resembles Pontoscolex. You might not be able to see, but here's some, these are the setae, the little bristles or hairs on the ends of, on the body. And these, they're large, but they're straight, and this one, they are forming sharp hooks. And so this is morphological differentiation within this nominal species. So Lees went on and did a bunch of work with the haplotype using 16S and cytochromoxidase 1, got lots of different populations sampled, and got this haplotype network. And so in this case, the colors refer to the sites at which these things occurred, and then she's differentiated several lineages within this group. So the point of this is, with relatively simple technology, like getting out a couple of gene fragments, you can learn a lot about the diversity of Pontoscolex, and then you can start asking, well, okay, so what's going on? Where did these Mexican populations come from? And here's the things near the Guianan Shield. And then Pontoscolex actually was described from Santa Catarina near Joinville by a guy in 1857. So even by then, after roughly 300 years of colonization by humans, it had gotten from the presumed homeland to Santa Catarina and was, he said, the most abundant earthworm present in every piece of soil. So those are the 3 or 4 lineages defined by these mitochondrial haplotypes. And she looked at them also using AFLP markers, and that confirmed basically that these lineages were, the nuclear genomes were distinct as well. Again, the colors refer to the sites of collection. So there's not just those 4, but there's a bunch of others that were not distinguished by the haplotypes of mitochondria. So again, AFLP, relatively simple technology, but more involved, I guess, than doing just simple barcoding, DNA barcoding. And then looking again at this sort of, where are these things from? And this is a very simple, old-fashioned clustering procedure that Lees and her lab used. They found, okay, here is a group from the north or northern Amazon basin near where we think that this worm is coming from, and that kind of helps a little bit confirm that we think, okay, what we thought about it. Lalopé, Gabon, there's, like I say, this worm is everywhere, and these guys are showing that there's a lot of movement of material into Mexico. So, and the conclusion then about Pontoscolex, that there's at least 4 lineages, perhaps more. There appears to be some morphological differentiation, but it's not absolutely clear that that corresponds to the genetically detectable lineages. And what we would like to do is cover more of Brazil. Remember, there's only a very few locations sampled in Brazil for this worm in this study. We could ask questions, are these different lineages some of them greater invasibility, some of them less. How important is it which lineage is in your area? Which lineages are most likely to be found in places that have not yet been sampled? Is there, look more into the ploidy level? There's a whole bunch of different questions still outstanding about this. So the great thing about Pontoscolex is that anybody in Brazil can probably take a little trip outside and find it. And if you live in an apartment building, okay, that might be a little bit more difficult, but you don't have to go far into some garden or into the countryside and start looking. You will find Pontoscolex. So what we're hoping to do is engage people in a little bit of a citizen science crowdsourcing thing to get a larger coverage of Brazil for Pontoscolex orithurus. And this relates to another thing, which is the Terra Preta, or Amazon Dark Earths project, which we're also working with. Okay, so now something to completely shift to a worm which is found only in southern and southeastern Brazil. It has a lot of small-scale regional or very regional variation, as you'll see shortly. And this is Urobanus brasiliensis. It was described in 1886 from a site in Rio de Janeiro in the Parque Nacional Serra das Orgãos at 2,000 meters of elevation. So that's what this one looks like, at least some of them. This sort of general, sort of very generic earthworm kind of appearance. They're typically sort of a gray-brown back. They may be a little different colors. Some are lighter, some are darker. And we go around and we collect and we see, oh, that's an Urobanus brasiliensis, and that's another one, that's another one. And we just keep getting them from many different locations. And then we started barcoding. And whoa, look at that. So lost the scale on this illustration, but these are deep divergences. This is somewhere in the neighborhood of roughly 15 to 20% divergence within the cytochrome oxidase 1 barcode region of 658 bases. Any short branches, these are all from the same spot or very close. So we started to ask, okay, well, what can we do with more Urobanus? And maybe we can start looking into species delimitation questions. And what about other markers? So we expanded the sampling. We started looking at, I started looking at the morphological characteristics. We wanted to get DNA barcodes for everybody in AFLP genotyping for all of them. And that was the first round. And then we compared those clusters we generated from barcodes as with those we generated from AFLPs. And then tried to integrate genetic and morphological data, which failed because the morphological data told us nothing. The type locality is here. Most of our sampling has been in sort of from here down in through all the way into the near Pelotas in southern Rio Grande do Sul. This is briefly the analysis of the barcode data. We used a Bayesian, we used Mr. Bayes with suitable models and defined each codon as a partition, and then we rooted with a different Urobanus species from French Guiana and another genus, Andiorhinus, for which we had a number of individuals. And then with the AFLP, we can get a broad sampling of the nuclear genome and do that relatively inexpensively for an organism that does not have genomic resources or any relatives that have it. And so in morphological data, I scored a bunch of sort of standard, relatively easily measured and observed characteristics inside and out. So moving right along, AFLP genotype clusters here with color coding for regions that we later identified. So each tip is a sample in this case. And so we have a northern coastal group, and we have a southern coastal, and then northern inland, a couple of those clumps, and then southern inland, and I'll show you what I mean by those. The barcode data, there's as many terminals in this as here. However, they sort of disappeared in the forming of this unrooted graph. And so we have, again, the northern coastal, the southern coastal, northern inland, and southern inland. Roughly the same layout.